Okay, I think, look, I'll make a start, and the meat of the thing will come with Kwang's presentation in a few minutes anyway. But thank you so much to everyone for joining us for this next exciting seminar in our exciting seminar series. You'll be well aware of who we are now by now as a company, Exciting Instruments. We lean into the puns, it's the only way. And so I'll talk a bit about Kwang in a minute, but just a little bit about Exciting Instruments. And as I'm sure many of you are aware, we make exciting instruments that excite molecules with laser light, hence the slightly punny name. And what those instruments do, this is our V1, the EI Flex, for which Kwang was one of our first early adopters, actually. And this is an instrument that sits on the benchtop. It doesn't need to be in a laser-controlled environment. It can sit on ambient light conditions even next to a floor-to-ceiling window. One of these that we've actually put in place, for example, in the University of Copenhagen. And it enables you to do in-solution single molecule FRET measurements and fluorescence correlation spectroscopy and cross-correlation spectroscopy. So this allows you to do things that very much like Kwang's going to talk about, where you've got static structures maybe of the things that you're interested in, sort of maybe in different conformations, for example, dimers, homodimers or heterodimers that exist in different shapes. And you may well have the static information from crystallography, from cryo-EM and other types of methods. But actually, it's the actual, what's really important is what are the actual conformations adopted in solution? How do they change upon interaction with substrates, with ligands, with cofactors, so nucleotides in the case of Kwang's work here. But then, how do they, how do they actually move, right? Can you actually obtain the rates of interconversion and when they're interconverting and when they aren't? Because actually it's that dynamic piece here that turns out to be critical for function. And so I think one of the themes we're going to try and draw out of today is that, structure-function relationships have been known, that paradigm has been key for a long time. And it's not wrong, but it's not the full story. And I think that's really, really important, that actually you need to insert dynamics there. And it's structure dynamics function that's actually closer to the truth, or at least one step closer to the truth. So look, just a quick overview of where we've come now as Exciting Instruments. It's been a year since we took on our £4 million seed funding, sort of 5.2 million US. We've now grown to sort of 16 people across the globe. We've got Audrey, who's just on just now. It's just started in West US, so San Fran sort of area. So super excited to have her on board. Jake is on the call as well, who's just started in Boston a few months before that. Roman was started in Dresden in Germany. So we really are now global in terms of all of our, but in fact, even with boots on the ground, if you will. We're super excited about the Flex, obviously, and that's now in many labs around the world, across the US, Europe, and the UK, enabling people to do those single molecule FRET and FCS experiments. But we're particularly excited about the launch of our new high throughput instrument. So this is something that we've developed in-house now over the last 6 to 12 months. It's fully automated, so there is no pinhole adjustment. There's nothing for you to do except put your plate in, click go, and print your nature paper. So even easier than it was before. But no, it literally can do single spot, so on a cover slip, but also 96 well and 384 well plates. So with that automated acquisition analysis, we've got a bunch of new patents coming out there, and actually some specific assays which we're developing for this in the molecular glues and PROTAC space, in protein aggregation, and in antibody QC. And so there's some really nice work coming out there. And in fact, some of that with Lawrence Livermore National Lab, there's an app note coming out joint with them. We'll show that very soon, and a bunch of other bits with various industrial partners here. But that's enough about us as a company. This is really about Professor at UMass Chan, where he's been now for 5 or 6 years. Is that right? Yeah, something like that. It is 6. Wow, time really does fly, doesn't it? So 6 years there. Quang received his bachelor's from the University of Technology, Science and Technology in China, and then went on to get a PhD in chemical engineering at Caltech before a couple of different postdoc positions, and then starting his lab at UMass Chan. And Quang is an absolute expert in single molecule things prior to this, but I think that this was his first foray into solution-based measurements, and so it's great to have Quang with us, and I will happily hand over to him now and let him share his screen to take us through the great work that he's been doing on these amino acid sensing system, the GTPases heterodimer.
All right, well, thank you very much, Tim, for the very kind introduction, and thank you for having me here. It's a great pleasure to share some of my work on the single molecule FRET study on this very unique GTPase dimer within this nutrient sensing system. So we all know that amino acid is one of the basic building blocks of all living organisms. So they can form the backbone of proteins, they can participate in numerous metabolic reactions, and they can serve as neurotransmitters. So considering their importance, a cell should coordinate its survival strategy based on the availability of an amino acid in the environment. So in the presence of amino acid, they should start to take in all these nutrients and start to grow and to proliferate. However, in the absence of amino acid, they should start to eliminate all these energy-consuming processes and sustain the most fundamental process of the cell in order to survive this difficult time. So how does eukaryotic cell sense amino acid availability? It turns out the answer to this question is tightly linked to this protein kinase complex called mTOR1, or mechanistic target of rapamycin complex 1. This is the protein kinase complex. It will phosphorylate its downstream targets to carry out its function. In the absence of nutrients, mTOR1 is inactivated and it resides in the cytosol. However, in the presence of nutrients, it will translocate to the lysosomal surface, where the kinase activity is turned on. Upon activation, it will start to boost up mRNA translation, it will inhibit autophagy, and will boost up ribosomal biogenesis. And all these downstream targets will start to stimulate cell growth and proliferation. Then how does mTOR1 get turned on? There are two GTPase units that's regulating this process. The first one is the Rag GTPase heterodimer, which we'll be talking about today. So the Rag GTPase heterodimer consists of two subunits, Rag A tightly bound to Rag C, and it responds to amino acid signal. So in the presence of amino acid, the Rag GTPase will recruit mTOR1 from the cytosol to the lysosome surface to complete this translocation. And on the right-hand side is the Reb GTPase, and it will respond to glucose signal. So in the case of growth factor and glucose signaling, the Reb GTPase will trigger internal conformational changes within mTOR1 to turn on the kinase activity. So these two GTPase units form a coincidental detector to ensure that the cell will only grow when you have the basic building block, which is amino acid, and when you have the energy, which is glucose. So at this moment, none of these protein complexes directly touches amino acid, but instead, a suite of protein complexes have been evolved to directly sense amino acid or transduce the amino acid signal. For example, arginine in the cytosol is sensed by this COST1 protein, and the signal is relayed to the Rag GTPase through this GATA2 protein complex. On the other hand, arginine within the lysosome is sensed by this SLC38A9, an amino acid transporter. It directly binds to the Rag GTPase to modulate its function. So, as you can imagine, because this is an important pathway in the eukaryotic cells, mutation within this pathway have been tightly associated with all the diseases, including abnormal cell growth like cancer. For us, we are very interested in how to study how these proteins transmit the amino acid signal at the molecular level, and that is the focus of my lab. So we decided to start with a Rag GTPase heterodimer because of several reasons. First, it directly recruits mTOR1 to lysosome surface, so it's occupying a key position in this critical biological process. And second, it seems to be where all the amino acid signals converge to at this moment. And third, it has a very unique architecture, which makes the biochemical property very interesting. So I will elaborate on this point in a minute. So just to make sure everyone's on the same page, so what is a GTPase and how does it do the function? So GTPase is called guanosine nucleotidase. It can either bind a GTP, guanosine triphosphate, or a GDP, guanosine diphosphate. And based on the identity of the bound nucleotide, it will display different conformations. So probably the most notorious GTPase that you have heard is this RAS GTPase. As you can see that in the presence of the GTP-bound form, the switch 1 region shown here in red and switch 2 region shown here in cyan will display a certain conformation. On the other hand, when it's bound to GDP, switch 1 and switch 2 region will show a different conformation. And these conformational changes means life and death. So it will turn on the pathway or turn off the pathway. In order to turn on the pathway, what you have to do is to open up the nuclear binding pocket to release the bound GDP. Because cellular concentration of GTP is 10 times higher than that of GDP, a new GTP molecule is likely to rebind and activate the pathway. However, if you want to turn off the pathway, all you have to do is to hydrolyze a triphosphate to generate a diphosphate, and that will turn off the pathway. So we can consider this as a molecular switch inside of the cell that precisely controls a certain biological process. So in the case of wild-type GTPases, the nuclear loading state, the conformational state, and the functional states, these three states are totally coupled. You bind to either GTP or GDP, you display a certain conformation, and you carry out a certain function. But when you have mutations within that GTPase, for example, there's a lot of mutants in the RAS GTPase that's been found in cancer, you have decoupling of these three states. And we will talk a little bit of this in today's presentation. So in the case of normal GTPase, like regular GTPase, signaling GTPase, you only have one GTPase unit to worry about. It combines GTP or GDP, and will show conformation and carry out its function. But in our case, the Rag GTPase has a unique architecture because each Rag GTPase functional unit always contains two subunits. Instead, besides nuclear binding domain on the N-terminus of Rag A and Rag C, they each contain a roadblock domain at the C-terminus that tightly dimerize together. Since the discovery of this Rag GTPase heterodimer, its behavior is very interesting. And here is the first piece of result we're going to see. Since the discovery of this GTPase, the nuclear loading state has been very non-canonical, to put it at least. So in this case, we're looking at the interaction between Rag GTPase and its downstream effector, which is mTOR1. It will recruit mTOR1 to lysosome surface. So we are just doing a very simple co-IP experiment to probe the interaction between the Rag GTPase and mTOR1. If we transfect the cells with wild-type Rag A and Rag C, we can see that a certain amount of mTOR1, here shown by the two subunits, Raptor and mTOR, is associated with the Rag GTPase. If we introduce point mutations on this Rag GTPase to force both Rag A and Rag C to bind GTP, we can see that there are some changes, but really not a lot. And so is the case when we force Rag A and Rag C to bind GDP. We observe a very strong phenotype if we force Rag A and Rag C to bind to opposite quote-unquote nuclear loading state. In the case of Rag A bound to GDP, a diphosphate, and Rag C binds a GTP triphosphate, we can see that it loses binding affinity to mTOR1 and pulls down a much smaller amount of mTOR1. However, when Rag A binds GTP and Rag C binds GDP, it strongly interacts with mTOR1 and pulls down a lot of this complex. So The behavior of this is very interesting, and our question here is like, how does it do that? So when we first started to study this protein, this is the first question we asked. We did a lot of biochemistry and tried to figure out the molecular mechanism, and I wouldn't bother you with all the details, but I want to show you one piece of information on the GTP hydrolysis by the Rag GTPases that we did. So to study the GTP hydrolysis by the rags, we carry out the reaction actually in two different ways, and that's the single turnover versus multiple turnover reaction. So in the case of single turnover, we put in a very small amount of hot GTP and saturate this reaction with increasing concentration of Rag GTPase. Because each rag contains two subunits, so at this moment, only one subunit will have one chance to hydrolyze one round of GTP, and that's why we call it a single turnover reaction. And this mimics this condition where rag is singly loaded with one GTP molecule. On the other hand, if we do this reaction on the other using another setup called multiple turnover reaction, here we maintain the Rag GTPase at a relatively low concentration and then saturate this reaction with increasing concentration of GTP. In this case, we can force both Rag subunits to bind GTP and hydrolyze GTP, and this will mimic the dual GTP loaded nuclear loading configuration. When we carry out this experiment, what we first see here is we do that in a single molecule, in the single turnover condition, the Km, which is the binding affinity of the GTP to the Rag GTP, is very tight, which is 5 nanomolar. So this is similar to all the other GTPases, other signaling GTPases, which has a tight binding affinity to the GTPase, to the nucleotide. And the Kcat is very slow. It's 2 × 10⁻⁴ per minute. However, when we do it in a multiple turnover situation, we see that the Km value increased by 1,000-fold. That means the second GTP should bind to the remaining subunit at a very weak binding affinity. So this means the Rag is discriminating between the pre-bound and the later bound GTP. And the later bound GTP will be hydrolyzed at a much faster rate, like 10 to 15-fold, than in the single turnover condition. So this will tell us how the Rag GTPase work, and we come up with this locking mechanism to figure out how the Rag GTPase maintain the single GTP loaded configuration. And now it's a question, how about the conformational state? We start to correlate the nuclear loading state with a functional state, but we're missing one link, which is the conformation of this Rag GTPase heterodimer when it's carried out its function. So we need to define this a little bit better here. So for the Rag GTPase, we have to define the local conformation from the global conformation. In the case of local conformation, we mean the conformation of the nucleotide binding domain in response to different nucleotides. In this case, we know this pretty well because the Rag GTPase belongs to the off family GTPases. And in the case of GTP-bound configuration, switch 1 region will swing to the top of the nucleotide binding pocket, forming a lid. However, in the case of GDP-bound, switch 1 is relaxed and extend towards the other direction. So this is the local conformational change, and we know pretty well. However, the global conformation, which is the relative positioning between the two nucleotide binding domains, this is very unique to the Rag GTPase. And over the years, when people carry out structure studies of this GTPase, people observe all kinds of conformations here. For example, in the first column, when Rag A is binding to GTP and Rag C binds to GDP, these two nuclear binding domains, they rotate towards each other and form a close conformation. However, in the second column, if both Rag A and Rag C binds GMP-PMP, which is a non-hydrolyzable GTP analog, you can see that the Rag C's nuclear binding domain start to swing out and form a more open conformation. So what we want to ask this question is, does the global conformation really matter in this case, specifically in the Rag GTPase case? Because we know local conformation does matter. So does the global conformation also matter? To do that, we have to go to single molecule FRET, which is our favorite method here. So in order to do single molecule FRET, first we have to label the Rag GTPases. Native Rag GTPases contain multiple, like 14 different cysteines. So we can't just directly use melamine chemistry to label this protein. Otherwise, it will light up like a Christmas tree. And there's no way to get into site-specific label. So first, we have to generate a version of the Rag GTPase that doesn't contain that many cysteine residues. So we mutate the cysteine residues to generate a cis-like version of the Rag GTPase. As you can see here, in lane 4, it doesn't get labeled by melamide conjugated dyes. So we can introduce artificial cysteines either on Rag A or on Rag C side to get site-specific label at our desired position. Of course, we have to make sure that the cis-like version of the Rag GTPase behaves exactly like wild-type Rags. And what we did, we measured the binding affinity to nucleotides, we measured the binding affinity to upstream regulators or downstream effectors, and we measured the hydrolysis rate. And this cis-like version of the Rag GTPase behaves almost identical to the wild-type Rag GTPases. And we did one more thing, which is to use the cell-based assay to see that if the cis-like version of the Rag GTPase can still mediate amino acid signaling in knockout cells. So for example, here we're looking at the phosphorylation of S6K as a readout for mTOR1 activity. In the wild-type HEK293 T cells, in the absence of amino acid, you can see that the phosphorylation is down, while in the presence of amino acid, the phosphorylation is up. That means it's being regulated by amino acid. The TOR activity is regulated by amino acid. If you knock out Rag A and Rag B, like if you knock out the endogenous protein, the cells pretty much becomes like a zombie state. It doesn't respond well to amino acid withdrawal or supplementation. While we reintroduce wild-type Rag A, we can restore the sensitivity to amino acid, and so is the cis-like version of the Rags, or the cis-like version that we engineered an artificial cysteine on that. So this supports our hypothesis that cis-like version of the Rag behaves almost identically to wild-type Rags. And then with this cis-like version in hand, we can start to put dyes at our desired position in order to track the conformational change. We introduce the E98 on Rag A. We mutate that to a cysteine, and we mutate M151 to a cysteine, and we label these two with Cy3 and Cy5 dyes. The alpha carbons between these two residues is around the distance between the alpha carbon is around 43 angstrom, so which is the sensitive region for Cy3 Cy5 dye pair. So if there's any conformational distance change between these two nuclear binding domains, we should be able to track it. And then we have to use our favorite method, which is alternating laser excitation based single molecule FRET system. So here we label the Rag A, we diluted Rag A, Rag C, and diluted to a very low concentration, and then use the confocal laser beam to excite these molecules and record the response of photons. So here is the raw data. We excite this molecule diffusing through the laser beam. You can record the donor and acceptor emission at the corresponding PMT. And then with this photon stream, what we can do is we can analyze the FRET efficiency, we can calculate stoichiometry between the dyes, we can do the burst variance analysis, and then we can use hidden Markov modeling to extract the hidden FRET state within these photon streams. And what we will see here is a typical readout. There are two parameters that's most important here. The first one is FRET efficiency, which represent what was the distance between these two dyes. And the other one is stoichiometry, which is the relative ratio between these two dyes. Because of the difficulty in the labeling condition, so Alex is particularly useful in this case. The reason is is actually is actually pretty simple because the Rag GTPase, what we cannot do is to purify Rag A and Rag C separately, label those, and then combine them together. Of course, this is the ideal situation, but we can't do that. The reason is because at the interface between Rag A and Rag C, there's a lot of hydrophobic residues. If we only express one of the subunit, it will not get, it will just crash out. There will be no active proteins. So we have to co-purify Rag A and Rag C in this heterodimer and put them down. So in this case, we cannot do the site-specific, we cannot do the one-to-one like differential labeling. We have to do the stochastic labeling. So inevitably, we will have a molecule that contains 2 donor dyes or 2 acceptor dyes. So in this case, Alex will be able to tell us the difference between donor-only population, acceptor-only population, and the desired population, which has only 1 donor dye and only 1 acceptor dye at a stoichiometry between 0.5. So this is the ideal situation and a very ideal technique for us to use. And then we can do the experiment. The first experiment we did is we use EDTA to strip away all the bound nucleotide and only has the Rag GTPase in the aphel state. Okay. So in this case, none of the Rag A or Rag C has any nucleotide bound, so they cannot talk to each other. In this case, what you can see here is in a FRET histogram, you see a dominant low FRET population. That means the Rag A and Rag C nucleotide binding domains, they are far away from each other. They are not talking to each other at all. And then what we do is we put in GDP, guanosine diphosphate, to force both Rag A and Rag C to bind GDP. In this case, you can see that a high FRET state starts to show up, and there's a medium FRET state to also start to pop out. But still, the majority of the molecule is still at a low FRET population, which means the 2 nucleotide binding domains, they are still far apart. And then we can add in the GTP molecule. In this case, we have 1 micromolar of GTP to control that only 1 subunit is loaded with GTP, and the high FRET state start to dominate in this case, suggesting the 2 nucleotide binding domain, they start to complicate. And this is the first time we can start to correlate the global conformational state with the nuclear loading state of this Rag GTPase heterodimer. And then how do we do the locking mechanism? So, well, through biochemistry, we know that when Rag GTPase is loaded with a single GTP, it's behaving differently when it's loaded with 2 GTPase, two GTP molecules. In this case, we have to introduce the burst variance analysis and Hidden-Markov modeling. So it will extract how dynamic conformation is when this molecule is diffusing through this confocal volume. So here is the raw data of the photon stream. If we calculate the FRET efficiency here, we can get 1 spot in the ES plot, as we have seen before. But that is an under-usage of this photon stream data. So what people have come up with a genius idea to make more use of this data is, instead of calculating the FRET value from all the photons here, what they did is they create a window, for example, 7 photons, and they calculate the FRET value of this subpopulation of photons. Okay, so this will give us a snapshot of this of the Rag GTPase when it's diffusing through this confocal volume, and then they create a sliding window and calculate the next snapshot, and then they can do this for all the photons in this entire burst. So the idea is quite simple, actually. If you only have 1 conformation, or if the 2 dyes are relatively static in when this molecule is going through the confocal volume, you will have a small variation when you calculate the standard deviation of all the subbursts here. But on the other hand, if the protein is bouncing around when it has multiple conformation, when you calculate using the sliding window, you will have a big variation. That's the idea. That's the genius idea by Kapanides group. And then we apply this idea to our system here and calculate burst variance analysis of the Rag GTPase under different conditions. Here you can see that in the apo state, we don't have any nuclear bound, and the Rag GTPase they fall apart from each other. And the burst variance analysis here, the triangle means the actual experimental data, and the semicircle is the theoretical limit. So the more difference you see between the experimental data and the theoretical limit, the more dynamic that population is. As you can see here in the mid and high FRET population here, there's a significant deviation from the theoretical value, suggesting that the medium and high FRET state here is not very stable. So they have a lot of dynamics inside, and the rags is really, really dense in here. And then what we can do here is we can compare the single GTP loaded configuration with a dual GTP loaded configuration. In the case of single GTP loaded configuration, when it reaches the high FRET state, the experimental value, the triangle, perfectly matches the theoretical value, which means that conformation is a very stable conformation. However, if we flood the Rag GTPase with the excess concentration of GTP, you can see that although it can still reach the high FRET state, but the high FRET state is much more dynamic than in the single GTP loaded configuration. So this is the locking mechanism that we observed in the biochemical study. When the rags is loaded with two GTP molecules, one of them has to be hydrolyzed, and this dynamics is what we have seen is its Rag GTPase is trying to hydrolyze one of the bound GTP molecules here. And then we can further use hidden Markov modeling to quantify the transition kinetics between the low, medium, and high FRET population, and consistent with our prediction. In the case of the dual GTP loaded configuration, the transition is much more dynamic in between these states, suggesting these two nuclear binding domains are talking to each other and negotiating which GTP has to be hydrolyzed. So now with this tool in hand, we can start to introduce so many things. We can do so many experiments. We can introduce point mutations to the Rag GTPase. We can use different nucleotides, and we can study the conformational change upon binding to upstream regulators and downstream effectors. For example, here is one example when the Rag GTPase is binding to mTOR1. If I can bring our attention back to this process. So when Rag GTPase is in the correct nuclear loading state, it will recruit mTOR1 from the cytosol to the lysosomal surface and fulfill this translocation process. Now, how does it change the conformation? Okay, so to do this experiment, we first incubate the downstream effector, which is Raptor, with the Rag GTPase, and pass through this mixture through a gel filtration column. So the Rag GTPase is detected at two different positions: at a high molecular weight position, which means it's bound to Raptor, and then a low molecular weight position, which is the unbound Rag GTPase. And then we can directly apply this eluate to the single molecule microscope and see the conformational change. And in the case of unbound Rag GTPase, consistent with our previous result, the high FRET state is dominant. However, binds to Raptor, the medium FRET population start to pop up, suggesting the conformational change is happening here, because we know in the case of the activated Rag GTPase is in the closed conformation, and when it binds to Raptor, something has to happen in order to open this up. So this brings up to the conclusion here, which is the Rag GTPase, well, when it's activated, it's in the closed conformation, the global conformation is closed. And at the end, the global conformation is open. And somehow, either by induced fit or in a conformational selection process, the Raptor is controlling the global conformation in order to be recruited to the lysosomal surface. And now we have to ask one last question. What's the biological relevance of this global conformation? Of course, the local conformation of a GTPase is definitely important, and so is the case in our system, the Rag GTPase. But is the global conformation, which is unique to the Rag GTPase, is the global conformation really important? So what we need here is to identify a separation of function mutant that doesn't change the local conformation, but modulate the global conformation. So how do we do that? And obviously, we can't touch anything on the nucleotide binding domain because that will couple the local changes to the global changes. So we have to extend our sight beyond the nucleotide binding domain at the interface between NBD and roadblock domain. And as we immediately found that there's a very conserved protein residue that mediates the connection between the NBD and the cRNA here. And from the structure studies, this protein residue seems to be very important because in the case of GDP-bound RAG-C, for example, this protein residue is flipping to the right-hand side and is directing the nucleotide binding domain of RAG-C towards the left-hand side. However, in the GTP-bound configuration, this protein is flipping to the left-hand side and it is directed NBD of RAG-C backwards. So it probably mimics how the Rag GTPase changes as global conformation. So what do we do about this protein residue? We mutate it. We mutate it to a glycine residue based on the Ramachandran plot because glycine residue is exploring a much wider conformational space than the protein residue. So in theory, that should be able to allow the nucleotide binding domain of the Rag GTPase to flip a little bit more at a wider conformational space than when it's connected to a protein residue. So we make these mutations, and the first thing we do is to make sure that we didn't disrupt the local conformation. We measure the binding affinity of these mutants to GTP and GDP and found they are very similar to wild-type Rag GTPase and are hydrolyzing GTP at a similar rate to wild-type GTPase, suggesting we didn't touch anything local. And then we put this mutant back in our single molecule system as a control. First, we always have to do the apo state, which is a negative control. We strip away all the bound nucleotide and found it doesn't matter when you have a mutation or it's wild type, the Rag GTPase mostly reside in a low FRET population. And then we put in the GTP and see what's happening here. In the case of wild type, the Rag GTPase can lock into the closed conformation very efficiently. However, if we do the mutation on Rag-A, we can see that the high FRET state almost completely vanished. So that means Rag-A cannot lock into the closed conformation anymore. And in the case, if we do the same mutation, similar mutation on the Rag-C side, what we can see is that the bandwidth of the high FRET state, instead of being a nice and sharp peak, it starts to broaden up. So that means the high FRET state here is no longer stable. So that, and we further confirm that with burst variance analysis and hidden Markov modeling. So that means, although in this case, the two nuclear binding domains can still reach the closed conformation, but it's not a stable closed conformation anymore. Instead, it's probably a cluster of conformations around that equilibrium position. So this means we did generate a mutant that specifically disrupt the global conformation. And lastly, we can answer this question, ask the question, is it really important inside of the cell? So we can check the amino acid signaling inside of the cell mediated by these mutants. As you can see that wild-type Rag-A, Rag-C can mediate the amino acid signaling with no distortion. However, if we introduce either on the Rag-A side, the P2G mutation, or on the Rag-C side, the P2G mutation, the amino acid signaling is significantly elevated, suggesting that these two mutations, they are disrupting the amino acid signal inside of the cell, which is consistent with our prediction. So I think I've shown you enough data at this time, so which will bring us to the conclusion. So here we use the exciting instrument to build a single molecule system to visualize the global conformation of the Rag GTPase dimer. And it turns out that in addition to the local conformation of the GTPase, each individual GTPase subunits, the global conformation or the overall shape of the Rag GTPase is important during amino acid signaling. So yeah, that's the major conclusion of our study, and all the work is done by my grad student, Mr. Dylan Doxey here, and I'm very happy to share my study, and thank you for having me here, and thank you all for your attention.
Fabulous, fabulous stuff there, Quang. Thanks so much for that. So we've had a few questions already, and so I think we'll just move into a sort of more immediate question and answer, and then I've got a little bit of a more sort of fireside chat sort of thing that we're going to have. But there's been a couple of questions in the chat already about the labeling strategy that you took, and some sort of thinking, nice that maybe it was a bit lucky that your cis mutant worked and was sort of functional. So can you maybe just sort of talk us through in a little bit more detail about that process? Did you mutate every cysteine, or were you able to do some sort of analysis that meant you didn't mutate them all? Did you mutate every cysteine to alanine, or were there other things that you had to do to win, a cis-light mutant?
Yeah, so this is what we spend like half of our time on. So it's very unfortunate, but in different protein system, you have to work around this problem. So our strategy is for the surface exposed cysteine, we basically mutated to a serine residue, which best mimic the similarity between the sulfur and oxygen, so that will
Yeah.
introduce the minimum disruption. And there are some cysteines that's partially buried, so that's kind of the most troublesome cysteine. So what we did is we first aligned the sequence from human to yeast and see what like in other organisms, is that cysteine still conserved? And luckily, most of them are not. And some of them mutated to methionine, some of them mutated to a leucine residue. So we just borrow the knowledge from evolution and mutate that cysteine to the corresponding residue in another species, Rag GTPase. And that get us lucky. So of course, it will require different trial and error. Not every version will work, but somehow you have to. It's just try. You just try.
Absolutely. So the answer is that actually the surface residues you mutated to serines because and that's the most conservative, as you say, because it's a single atom change from sulfur to oxygen, and they're in the same row of the periodic table, column of the period, yeah, whatever. I'm supposed to be a chemist. Who knew? Anyway, that's fine. But then the others, that's I think, a really important strategy. So you actually went back through the evolutionary tree, you did your blast alignment or whatever, and then looked and saw other residues that were used in those places and then mutated to those residues. Is that right?
Yes. Yes.
And some of those were leucine, some of those were
methionine. Yeah.
methionine. Yeah. So that's it. Did you mutate every serine in the end, or did you leave a subset because they were buried?
I think we mutate most of them, and some cysteines are really like buried in the core, so we didn't touch that.
Right, and then you did I think what was nice is then you did the sort of the mock labeling experiment where you did the same conditions for labeling, but showed you got very little labeling, so that
Yes,
was
exactly.
that was
That's exactly
consistent.
right.
Brilliant. Hopefully that covers the all of the questions that were there around the labeling. So Torsten Hugel also asks, how do you selectively force the RAG-C to bind GTP and the RAG-A to bind the GDP in the dimer, or vice versa?
So there are two strategies here. The first is what we just use wild type, and what we can see is an average, ensemble average of half RAG-A half RAG-A loaded with GTP, or half RAG-C loaded with GTP. And the second strategy is we can always introduce point mutations that blocks GTP binding but not GDP binding. In this case, we borrow knowledge from the previous study on the RAS GTPase, like I can't remember the residue number, but there are specific mutations in the GTPase nucleotide binding pocket that you can mutate to switch off GTP binding but not GDP binding. In that case, you can have a homogeneous population of TD or DT state.
Brilliant. Torsten, did you want to turn your mic on and come back on any of that?
So, yeah, thank you very much for the explanation. I mean, this was very well explained, and the questions were well answered. Yeah, I think that's
Thanks.
That's fine. I mean, you just, yeah, I somehow missed that you used mutations to selectively bind GTP and GDP. Yeah, that's good if you have such mutations.
Yeah. Not always possible, right? So
Yeah.
brilliant. Thanks.
Maybe then the other question. So thanks. Yeah, it
Yeah,
was
carry
a
on.
great talk and great presentation. So for the hidden Markov model, you need to input the number of states. So how did you decide on this? I think you used 3 states, or you just said, okay, 3 is enough, or is there
So
any more
yeah,
behind?
so one idea is to also use the ICL integrated completed likelihood to measure. Like if you give it one more state, do you get more information?
Mm.
So then we calculate like based on we use
AIC or the BIC. Okay.
ICL and
Mhm.
find the optimal number of states. So that's how we decide. Yeah.
Okay. Thank you. Yeah.
Brilliant. Thanks, Torsten. So there's a couple more questions around the that was Torsten's second question. Then around the labeling strategy. What would you have done, Quang, if it hadn't been the same activity, right, with the cysteine mutants? What other strategies could you have employed to get site-specific labeling for single molecule FRET?
Yeah, so if this unfortunately didn't work, what we'll have to do is either do a tag-based labeling. For example, there's a SFP, that's called YBBR tag. This
Yeah.
is a tag that has like 11 amino acid that you can insert in the protein sequence. And you have to make sure that this 11 amino acid can form an alpha helix. So probably give it a couple of amino acid at the end of C-terminus of this small alpha helix. And once you have that insertion in your protein, what you can do is to use SFP protein to conjugate a version of the dye, which I think is CoA.
It's the CoA version of the
Yeah,
dye.
it's the
Exactly
CoA
that.
version of the dye into that sequence to
Yeah.
generate a labeled version. And that is very, very specific. And
Yeah.
you can almost get one-to-one stoichiometry label. So
Very
this,
much so. We've had many of our customers, users have had a lot of success with the YBBR approach, both at N and C termini of proteins, but also, as you indicate, actually in even in internal positions, this can
yeah.
work well.
Yeah, the second one I can recommend is to use the AMBER codon system, and all you have to do is to use AMBER express a tRNA that recognize the AMBER codon. So supposedly it's a stop codon, but that tRNA can recognize the AMBER codon and incorporate a non-natural amino acid, which is the bi-labeled amino acid, into that particular position of your protein. Of course, you sacrifice the yield by a lot, but well, you get what you get.
Yeah, I guess one of the beauties, and this speaks to one of the other questions, is that you don't need very much material for the single molecule measurement, right? Just one of the questions there was, how much material are you using? Can you and how long the measurements were? So maybe you could just speak to that quickly.
Yeah, so the amount of protein every time, so so we can do so every time we thaw a tube of protein, the most amount of protein we use is to determine the concentration, actually.
Yeah.
So every time you use so little protein at picomolar concentration, labeled protein, and you can get a whole bunch, very like several thousand bursts, like comfortably. So
Yeah.
what we did is we take like 10-minute movies. Like we take 5 or 6 of them together and then bin them together into the big file and then use the script to process them. Because, of course, as you know, we first drop a drop of labeled protein on a cover slip. At first, you get a lot of signal, but then as time goes by, the labeled protein gets stuck, get trapped onto the cover slip. So you're losing a lot of signal. At the end of this 10 minutes, you probably only got like 20% of the signal as we first started this movie collection. So what we did just a very simple strategy. We just at the end of this 10 minutes, we stop the movie and then we drop a new drop and then we collect a lot more signals. So
That,
yeah.
Yeah, that's great. So somewhere typically, I guess, yeah, you would use somewhere between 10 and 50 picomolar of your labeled material. Is that similar to you
Um,
guys?
yeah, I think we used a slightly more, like 50 picomolar to 100
Yeah,
picomolar, but
perfect. Yeah,
that's in the
perfect.
ballpark range. Yes.
Yeah, no, that's great. That's great. And then another one on the more on the labeling side, I guess there's a lot of people wanting to do these experiments and trying to get into the detail. So they're wondering if there were any issues in removing excess dye, or how you go about, I guess, once you've done your labeling, how do you remove the dye from the system? Because you definitely don't want that floating around in your single molecule experiment.
Oh, absolutely. So when we do the labeling, we put in like a protein concentration at 1 and a dye concentration at 3 or 5. So a little bit
Yeah.
extra dyes. So in order to remove all these dyes, we either use a gel filtration column on the FPLC, or we can hand pack a gel filtration column just by I think is super I forgot the brand. It's Superdex 10 like resin. We can pack
Yeah.
in one column, and then we can apply the protein to the top and separate the dyes. It's really easy because you will see two bands coming down the gel filtration column. The first band is a labeled protein, and the later band is the excess dye. And then you just collect the first band. That's your protein label.
Yeah, brilliant. And I mean we've had a lot of success with even just small spin
Ah,
solution columns actually as well. So
yeah.
there's a number of brands available. The one beginning with Z, which absolutely eludes me right now. Zaber, I think, is the thing. Yeah, I'm trying to look to my app specialists, but they're kind of in a different room and not looking at me.
Yeah, probably.
But yeah, so
Like,
those even the simple spin
like
columns.
like pre-packed column, yeah, this
Yeah,
little
yeah,
column.
exactly. And actually, one of the things that we're now doing as part through the company is providing some labeling kits
Ah,
with the dyes, with the buffer, and with the columns that we'll use. Because exactly that, then you can spin through the protein will come through in the eluent because it's bigger, and the free dye actually remains in the column. The beauty of these things as well is actually if you want slightly better separation, you just run them 3 times, sort of in series. So you just spin one, spin another, and then you're done. And so in our kits, that's exactly what you'll get. So I think that really helps. Awesome. Well, look, in the last kind of 5 minutes, I'd like to kind of bring this around to sort of a broader sort of question. Which is kind of where we started, Quang, as well, from the science side, which is, where are we in the whole structure function paradigm right now, right? What does this tell us about, are dynamics the missing piece, or are there other things even beyond that we're going to need?
yeah, so as you can imagine, like with the AI coming into the space, the structure field is done. Yeah, so in this moment, what AI cannot predict is the interaction, how fast the protein interact, and the specificity of everything here. So the dynamics is the missing factor here. Um, um, it's definitely more like if you have a complicated system, probably you will have to a lot of conformations to probe. So in that case, definitely go to the uh, to definitely use the method that can tell you the conformation. Structure studies will give you snapshots, and AlphaFold is doing incredibly in predicting snapshots, but it cannot predict like how like the populations of individual population. Some of the rare population could be really important. So in this case, I think we will still have our job if we study conformations.
Let's hope so. No, absolutely. I completely agree. And then just in the last few minutes, Quang, so talk us through because I think you've done some turf measurements before, right? So what was your thinking around, why you wanted to do solution-based measurements, and then what was it that led you towards the flex and exciting instruments as the right solution for that problem?
Yeah, the solution base is definitely advantageous because it doesn't require you to tether a protein to the surface, which will give you like a lot of artifacts sometimes. Some proteins, if you have a DNA molecule, like DNA, nucleotide molecules, it's such a blessing. So it will behave pretty well on the cover slip, passivated by PEG or some other reagents. But if you have a protein, sometimes they don't like to be tethered too close to a web of hydrophobic stuff. So they will like to crash out on the surface. And in the case of solution base, you just put a protein and let them be in the solution where it's most comfortable. And then you're watching the native conformation of these protein molecules. In a sense, this is definitely the advantage relative to the turf-based system. And also one other thing is it's much how do I say, it's much easier and because all you have to do is to prepare your protein, label your protein, and dilute them, and then apply it. You can get the answer in 10 minutes or so. While back then, when I was doing the turf-based system, it's like on the order of days or weeks.
We've been there, yeah. No, that's great. And then what was it? What's your experience been? I guess you've talked a bit about this of using the Flex, and Dylan, you're in your lab who's been using it. How have you found using our instrument?
Oh yeah, it's a great instrument. Yeah, definitely easy to use, and you don't have to buy like every piece of laser equipment and get and get your school involved in certifying all of those and make sure you're not shooting a 1-watt laser into your eyes. Yeah, so
No, that's right, because it's a class 1 laser product. Like for those of us who are old enough to remember CD players, and for the rest of you, it's your PS5. Absolutely. Well, that's absolutely brilliant, Quang. I really appreciate the time that you've taken to talk with us all about your great work on this space and about your interaction with us as a company as well. With that, I just thank everybody for joining us. This will be available, of course, on our YouTube channel and through our website after the event. And I promise Quang we're going to make him an influencer and a YouTube sensation. So, don't let me down, guys. But we really appreciate your time and appreciate we're at top of the hour now. As ever, if there's anything that you'd like to reach out to about this work, either email Quang directly or come via us. And if anyone's interested in talking about how they can do their own single molecule experiments on their own benchtop instrument, then of course do reach out. And Quang as said before, we'd be very happy to recommend and talk, to anyone around, his experience of the instrument, and of course reach out to us, and we're more than happy to demo and even work through specific pieces of work with you. We do currently have a competition running, which I will just let you know, which is to win 2 months of a Flex in your own lab. And so it's a 300-word, you can get ChatGTP to write the application, and many people already have. I can tell there's a lot of M dashes in there, but no. So do look at that. That's on our LinkedIn page, and we are keen to place a number of instruments into the labs doing the most exciting science. So if you've got a project that you'd really like to use a Flex on, well, you can send us samples, but you could also win the chance to actually have one in your lab for 2 months. So do get in touch. Really appreciate you all joining us, and thank you for your time, Quang, again. See you all next time. We actually have got the next 2 or 3 speakers lined up, so we'll be spamming everyone's email about that in the very near future. Thanks, guys. See you all soon. Bye-bye.